Hi, everyone. I'm Paula Harvey. I'm the education coordinator for Tejon Ranch Conservancy. And today we are going to do some really interesting counting and measuring and graphing. Let's take a look. So the title of this lesson is Biodiversity Inventory. And I don't know if you remember, way back at the beginning of all of these lessons, we did a compare and contrast exercise. And compare and contrast and zoom in, zoom out are the two most important concepts that you can do in journaling when you're doing all of those other lessons that we've gone over. And today we are going to be doing some comparing and contrasting of two interesting study areas where you'll be able to count the numbers, different numbers of species and the numbers of each species, and you'll be able to graph it. Now, there's two concepts I want to talk about. One is called species richness, and the other is species evenness. And so I want you to know that scientists will calculate species richness, that's the number of different species there are in a particular area, and then also the evenness, and that is the number of individuals of each of these species in a particular area. And they analyze these two concepts by creating graphs and by using computer programs and algebra and logarithms to determine the health or the stress of a particular area. What you're going to do is explore these patterns and ideas by using some simple math, some graphs, and look at two different study areas. You'll find this is really fun and interesting. So you're going to find two natural areas, and hopefully they're going to have lots of different things in them, like shrubs and trees and herbs, grasses, those types of things. And you're going to make those two study areas of the same size, and you're going to spend the same amount of time in each of those two areas. Now, the size of the area is going to depend on how many different species and how many of each of those different species you find in that area. So if you're finding an area that is very devoid of a lot of plant life, then your area is going to be larger that you're studying. If it's just full of stuff, then it's going to be a smaller area that you're studying. You're only going to want to spend about 45 minutes in each of these two areas. So you have to kind of determine the size of the area based on how much plant life there is. You're going to study each area, one for each day. And then on the third day, you're going to take that information that you have collected and you're going to create those graphs and plots. So this is a 3-day activity. So here's a summary of what you're going to be doing. You're going to make a biodiversity inventory of the species in the area. And so I highly recommend that when you are studying these two areas, they're close together. You'll notice when I show you my sample that I actually did one in a part of my yard and the other on the outside of my yard. And I found that despite the fact that they were right next to each other, they were extremely different. That was pretty interesting information that I learned from that. So you want to try and pick two areas that are relatively close together. You're going to record your ABCs. That's going to be what you notice with the questions and with your hypotheses and those connections this reminds me of. Then you're also going to do the 1, 2, 3s. This is the calculations that I'm going to show you how to do today. And you're going to do drawings, sketches, or diagrams. And some of those can be extremely easy. And in fact, I'll go into a little more about that later. And then you're going to count the number of each of those species, and you're going to use tally marks in your journal, and then you'll create graphs and plots from them so that you can analyze that species richness and species evenness that we were talking about. So here is the step-by-step procedure for doing it. You're going to take about 30 to 40 minutes for each of those study areas to record every kind of tree, shrub, and herb in that selected area. Now, you don't need to know what the names are. What you can do is sketch them and describe them in words and by counting, and then give them a letter as their identifying feature. And we'll go into that just a little more. All right, that quick sketch is going to be helpful so that you can go back if you need to, and you will be able to identify what it was you were studying. And then you want to use those ABCs, I noticed, to record that the most interesting details, such as the shape of the leaf, any fruits or flowers that may be on it, the form of that plant growth. Is it a tree? Is it a shrub? That type of thing. If you are drawing it, one of the fastest ways to do this, simply pluck a leaf off of that plant and trace it. Then you can draw in the veins of that leaf. Then you can color it and make sure those colors are really accurate to the tone of the leaf. If it's green, is it a gray green or a bright green? Try it and duplicate that. You're going to use tally marks to record the numbers of each of those species within that particular area. If there's too many of a species, you're going to have to try estimation. Now, again, set up those boundaries and make them about the same size, but do it before you even begin. So pick your locations and set up those boundaries. While you're journaling, I want you to consider these things. First of all, ecologists have found that ecosystems that have more diversity, that is more of different kinds of plants and animals, are more stable environments. Now, why might that be the case? Why is it that the more of different kinds of things would make an environment more stable? I want you to consider that while you're looking at your study site and trying to decide whether you think your site is more or less stable. And after you've determined that, think about how that area might change over the next 5 years and why might it change? What might be the causes of certain types of changes? Is it in a flood zone, for example? Is it going to experience drought? What might be the reasons for those changes? The other thing, again, going back to that compare and contrast, what are the similarities and what are the differences between those two study areas? Remember that when we first practiced this, we used a Venn diagram. You are welcome to use another Venn diagram to do a compare and contrast in addition to the other kinds of graphic organizers that you're going to create today. So here's mine. And I'm going to just show you how I broke this down so that you can consider doing that as well. First of all, the metadata is always there. First thing, before you even go outside, before you go to locate your place. Then, once you locate your study areas, identify what the size of it is. So I pasted off and came up with 26 by 15. I also wrote where it is. This is the east side of the house in the garden. Okay, and this area I identified as study area A. Here is study area B. So this is one journal entry that I did one day. And this journal entry I did the next day. Remember, this is going to be a 3-day activity. It's going to be 4 or 5 pages long. All right. Next, I trace those leaves and I provide a description of the leaf. So here's the description. I traced it, I put in the veins, and then I colored it. And notice each one is a little different color. Also, what I did besides just the leaf, I kind of was, for this particular one, I sketched how the whole plant looks, just really basic, not detailed, but just enough that if I was to look for it, I could see, all right, so the stem of this plant is alternating. And on each stem, there's several leaves. I would be able to do that simply by looking at that. Whereas looking at this is a very different kind of scrub, as is this. And that's really important. So I not only drew the leaf, but I drew a very quick sketch of what each of those plants looks like. All right, then I use tally marks to count up the number of each plant in the area. So see, all of these tally marks tell me there's 30 of this particular plant in this particular limited area, whereas this one has a whole lot more. Okay, and so I tallied all of those up. Now, these plants don't grow in straight rows, so you need to consider how are you going to make sure that when you're counting off all of these plants, you get it covered. So it's an accurate count. Are you going to walk back and forth? Are you going to count by standing there? How are you going to do it and get an accurate count? Plan that ahead. And then I labeled each plant in my study area. If you don't know what the name of the plant is, and you are not expected to know it, you simply get to label them. Here's species B, here's species A, here's species G, here's species I. So I just use letters to identify each of those plants. Don't need to know what the name of that plant is. Okay, so back to this idea of species richness and species evenness. The number of different species in an area is its richness. So when you've created what I showed you, you've created a species richness inventory where you recorded the different plants in the area. And then the next one, so when you do that, you're going to write down in your journal species richness at the very bottom of the page, and then write down the number of different species that you found. Now, species in evenness, also called the abundance data, is how many of each of those species it is. And so you're going to count up the total number of tally marks that you recorded for all of the species in that area, the total number. And at the very bottom of your page, you're going to write that number down and call that abundance data. All right. So once you've collected your evenness and richness data, it's time to go back indoors and start organizing this data so you can analyze it. So we're going to now go on day 3. All right. So give each species a letter code starting with A, B, C, and so on, just like I showed you on my journal page. And now we're going to use your diversity data to make a bar graph. On the horizontal axis, you're going to list each and every species, species A, B, C, and so on, starting with the plant with the most abundance. Now, that means it's not going to be in order A, B, C. It's going to be in the order of what's the highest number of plants. Okay. And then on the vertical axis, you're going to make a scale that goes up as high as the species with the largest numbers, the one that's the most abundant. So you may want to count by 5s or by 10s, depending on what your total numbers are. If your numbers are in the hundreds, you're definitely going to want to count by 10s to cover that. Then you're going to draw bars to show the number of individual plants for each of those letters A, B, C that you observe for each species. It's going to look something like this. First of all, notice the letters are not in order. It's not A, B, C, D, because letter G, which was a grass, was the most abundant right here. All right, so in this example, the graph was created in the order of abundance. Now, if you want to go back and label those A, B, C, D based on the most abundant, that's fine. You can do that, but you don't have to. It doesn't matter what order these letters are in. Okay. Notice also at the top of each bar, I put the percentage of coverage for each one. I'm going to show you how that's done in just a minute. It's just a little more information. It makes it a little more precise. Okay, so for this particular example, there were a total in area A of 510 total plants when I counted every plant of every species. All right, so out of 510 plants, plant G, there were 120 plants. So the way I calculated what this percentage was is I divide the smaller number by the bigger number. So 120 plants divided by a total number of plants, 510, came out to 23 hundredths or 23%. So what we're going to do is we're going to create a chart on the next slide that includes those percentages. So you'll be able to do your own bar graph very, very quickly. The chart's going to help you with that. And so what I did was I created that chart first. Well, I made the graph first, then I created the chart and I took those numbers and added it to the top of this graph after I created those. So you'll see how that goes. Okay, so the next thing you're going to create is called a rank abundance chart. So that's the top of my journal page is rank abundance chart. And I list my species, in this case in order. And I actually, what I did was I kept the letters going So area A was letters A through F. That's how many species there were in that area. And area B had 5 here. But you can do area A, B, C, D, and area B, A, B, C, D. Up to you how you want to do it. Either way works. I just put a space between and entitled area A and area B so that they were separated. But here's what I did. So here's species A. Species A, I counted 64 plants of that particular species. So I wrote that down. And then I wrote down species B and the number of plants, and species C and the number of plants, and so on. And then I added these numbers up and came up with my total number of species. And in this case, there were 359 total plants, adding all the different species. So now what I'm going to do is do my percentages. So in the third column, I take the 64, which was the first plant. Remember, when you're doing percentages, you're dividing the smaller number by the larger number. We always, when you first learned how to do division, you always divided the bigger number by the smaller number, but not when you're trying to get percentages. It's exactly the opposite. So plant A had 64 plants of that particular species. So I take that 64 and I write it here, divided by the total number of plants in the entire area, which is 359. And then I used my calculator and came up with 0.178. Now I'm rounding this up to 18%. It's 17.8%. 8 is 5 or more. And so you know when you learn to round in 4th grade that anything that was 5 or higher would round to the very next number. So that's what I did. 0.178 rounds to 0.18. Okay. And then after I've listed all of these plants down, all of the number of each of the plants, and divided each of those numbers by 359, I came up with my percentages. Then what I did in the next one is I just put these numbers in order. So the first one, letter E, had the largest number of plants, which was 28%. So that ranked as number 1. Number 2 had 73 plants and 20%. That ranked as number 2. So this is rankings of how common was each of these plants. Now I had to go on. So remember that I've talked to you before about the idea that once you think you finished your journal entry, you want to go beyond done. And so I always do go beyond done. So one of the things I did was identify the plants. And the second thing that I did was identified whether they were native plants or invasive plants. That is non-native, they don't belong here. And that because non-native plants will definitely affect the health of the native plants in an area. So those were kind of this particular area here. That's my go beyond done. You don't have to do that. All you have to do, is these 4 columns right here. So I did that for area A, but I also did it for area B. So here's my title, area B. Here's my letters I put them in order, but then I wrote down the number of each of these species and added all that and came up with 151 total plants. And then I divided the smaller number by the larger number and came up with percentages. And then I ranked them. This is the most common. Letter G was the most common. And then letter H was the second most common, and so on. Okay, so that's my ranking chart. And after I did that, I could take these percentages and going back, put them at the top of this chart that I've already made, this bar graph. Okay, that's how I did that. All right. Okay, next thing is to make this cool line graph. This just gets to look more scientific than ever. But we're really just taking that exact same information and we're just creating a different kind of graph. This one's a line graph instead of a bar graph. So this is going to help us to identify the abundance of each species to determine whether or not this is a healthy environment. So you're going to label the x-axis, this one at the bottom, you're going to label that ranking in abundance. That's right here. Okay, and then number it to the total highest number of species that you studied. Okay, so then remember, you're ranking them. The next one on the y-axis is the percentage of the sample. And we've already created that in that chart. You created those percentages. And that is the number that up to the highest percentage. And so in this case, one plant was 80%. So I had, or it was 78%. So I had to create this by tens, 10, 20, up to 80 in order to have enough room on that paper. All right. And then you're going to mark them. So for example, this particular plant was a high rating, but it was only 29%. And so I put that there. And this is area B and this is area A. So I made them different colors to make it easy, but I also labeled here so that whoever's looking at it, including you, will be able to know why it looks this way. Okay. And that's going to give us a lot of information about whether this area is healthy or not. Okay, so what did I do with all of this information? So area A, I ranked these plants. Here's the letters. Remember, you don't need to know the names of these plants. I put the names in, but you don't have to do that. Just put the letters and then ranking. You know, this was 100%. This is 100. This is 73. This is 64. These are the rankings and these are the percentages. And all together, it comes out to a total of 359. That's how many vinca did we find? About 100. This was an estimation. How many poppies did we find? About 73. When they got to the smaller numbers, these are more accurate numbers. But the 100, the 73, the lamb's quarter, I actually did count, but the poppies, I had to estimate that. And then what was the percentage of each of those? This number here, the total number all together when you add all of those species together. And I did the same with the area B. So here's the ranking, the letter identifiers for each of the plants. The rankings, this was 118 bunch grass, there were 19 rabbit bush, there were 10 buckwheat, and here's the percentages of each of those plants, and the total number is 151. Okay, so that's where all those numbers came from that ended up here. All right. And I've explained all of these things. I showed you that I've changed these colors so that it's easy to identify which is which. And then what I did was I wrote this definition down because I wanted to remember it. So the horizontal line on the graph shows high evenness lines that start out high and dip sharply. Low evenness is common in disturbed areas. All right, short lines that show low richness, fewer species, and the long lines are higher richness and show more species. Okay, so then I wrote my conclusions down. The area inside the yard has higher richness. That's area A, guys. That's this one that has kind of an even line. Has high richness and relatively high evenness. And then the area along the road, which was the other study area that was right next to it, it shows low richness, didn't have as many different kinds of plants. However, a lot of the native bunch grass that was there, I helped it to grow. And so it's really common. That's why this is such a big line. If I hadn't done that, this line would have been much smaller. But I did it because bunch grass is native and I wanted it to take over. The other thing is, though, that I removed the rabbit bush from there. So the number on rabbit bush may be lower than it would naturally have been. And then the buckwheat that grew there was the same. So really what I'm telling you in my conclusions, area B is a much more delicate habitat because one, I changed some things about it. And so it isn't as stable. Okay, let's talk a little more about drawing those conclusions. So on your graph, a horizontal line shows high evenness. That's this one right here. Okay, a vertical line shows low evenness right here. right there. That shows low evenness. It means there's a lot of one plant and not as many of the others. Okay. And in this area, area A is relatively even in number for each of the species. However, area B shows high number of bunch grass and the other numbers are low. This is what I just explained to you. Therefore, there's very low evenness in area B. This is common for an area that's often disturbed, which in fact, that's the case here. And in this case, it is the side of the road and it's frequently disturbed by all kinds of things. So now I'm going to write my conclusions down at the bottom and explain some of that there. And that's the last thing that you're going to do on your analysis. And that is it. It's really mathy, but it's really fun to take those numbers and play with them. Once you've collected those numbers, then think, how can I display those numbers? And what do those numbers tell me? And what are my new questions that I have now that I have those numbers? It's very, very scientific. I hope you had fun listening to this, and I hope you have lots of fun doing this project. So enjoy it. We have one more lesson in metrics and quantification, and I'll see you soon. Bye for now.